A pleasant day to you, icons and lady icons. Welcome to our discussion in this module, which is all about the central dogma of molecular genetics. Now, in this particular module, we are now actually going to apply all the discoveries and how they actually come up with the gene expression. Now, in the case of the central dogma of molecular genetics, we're actually going to focus on three major things. The first one, we're going to have the concept of replication. Then the second one, we are going to consider here the transcription. And lastly, it would be translation. And when it comes to the central dogma of molecular genetics, Francis Crick is actually the person who actually gave us this certain type of idea that the different types of proteins will simply be expressed based on the different DNA bases that is actually present in our body. And remember, in the case of the concept of the central dogma of molecular genetics, it is happening both on prokaryotic cells and eukaryotic cells. It is more complicated when it comes to the eukaryotic cells, unlike in the case of the prokaryotic cells, which is also discussed and presented on your modules. You can actually observe here that we actually going to start with the genetic material that is present in all of us, which is the DNA, followed by the presence of the mRNA that would be the product of transcription, and lastly, this mRNA will simply be translated to the different polypeptide genes that will now be transformed as proteins right after the process of the gene expression. Now, let's simply start with this particular concept. The synthesis of a new strand of both the DNA and even the RNA, it is actually running from the 5' to 3' direction. Always remember that. We actually have here three models of DNA replication, and it was actually evaluated by the study conducted by Mendelssohn and Stahl. We have here the presence of the conservative, semi-conservative, and the dispersive model. We can actually consider here that the DNA replication is an example of the semi-conservative model because only a strand of the DNA will be served as the template, then a new complementary strand will simply be created. And this new complementary strand, that would now be the new strand of the DNA that will be responsible now for its certain type of replication. So basically, it is not an example of a conservative and a dispersive model since those two types of DNA replication are not feasible. And once again, it was observed by Mendelssohn and Stahl that when it comes to the DNA replication, one strand remained to be the older strand while it would be considered as the template for the creation of the complementary strand, which will now be the new strand of the DNA. Okay? So that's the thing when it comes to DNA replication. It is an example of a semi-conservative model. And remember that replication is semi-discontinuous. It simply means that because of the 5' to 3' synthesis of the polymerase and the antiparallel nature of the DNA, it simply means that it is actually creating two different types of replication happening on two strands. It is either the leading strand or the lagging strand. Now, what is the difference between a leading and the lagging strand? In the case of the leading strand, it will only need one primase, which is the enzyme that is responsible for the initiation or for the start of the creation of the complementary bases that would be present on the leading strand. It will only need one primase. Then after that, the primers that would simply be present here, that would be the foundation for the creation of the different strands or the other parts of the leading strand. When it comes to the lagging strand, it is now the discontinuous part of the DNA replication since it is now actually possessing different types of segments and fragments known as the Okazaki fragments. In the case of the lagging strand, it is now utilizing a lot of primers and utilizing a lot of enzyme primase for it to have its complementary base. The reason why it is known as lagging strand because it is lagging. There are different gaps. It is discontinuous. While in the case of the leading strand, it is continuous. Now, speaking of the different types of enzymes that are actually present whenever we are considering here the presence of DNA replication, let's simply start with the presence of the helicase. When it comes to the helicase, it is actually one of the first enzymes that would be responsible for the initiation of the process of replication. The helicase, it is responsible for the unwinding of the double helix, or simply it is now responsible for opening up the double helix. Then, as I mentioned earlier, we have here the presence of the primase. The primase, it would be responsible for the synthesis of the RNA primers that would obviously lay the foundation of the start of the presence of the complementary strands that are present in this DNA strand, either on the lagging or the leading strand. Remember, when it comes to the leading strand, they are utilizing only one primase, but when it comes to the lagging strand, as you can observe here on the lower portion, there is actually the primase, and this part now is the lagging strand. It is lagging since there are different gaps. The one that is present on the upper portion, this is now the leading strand because it is just possessing continuous action of the polymerase. Then the next one, there's now the presence of the single-strand binding proteins. The single-strand binding proteins are responsible for the stabilization of the single-stranded regions, or there's actually the time in which this single-strand binding proteins would be responsible for cupping or creating a certain type of protection on the exposed hydrogen bonds in which no nucleotide would attach themselves to the free hydrogen bonds that are present on the exposed DNA template. After the presence of the single-strand binding protein, we now have here the presence of the DNA gyrase. The DNA gyrase, it is also the one that is responsible for initiation because it is responsible for relieving the torque, or it is now responsible for straightening and removing the twist that is present on those DNA strands. Remember that the DNA, it is a double helix structure and it is twisted. So the DNA gyrase is responsible for the release and the removal of the torque and straightening of the DNA strand. Then followed by DNA polymerase III. DNA polymerase III, it is now responsible for the creation of the DNA strands, the complementary DNA strands. DNA polymerase III, it is responsible for the creation of the longer strands. DNA polymerase I, on the other hand, it will now create DNA polymers also, and they will erase the primer. Since the primer, it is in the form of RNA, the RNA that is present on the primer would simply be removed and it will now be transformed to DNA polymers. Specifically, if there is the presence of uracil when it comes to the primer, it would be removed and transformed to DNA, which is its counterpart is thymine. Okay? Remember, when it comes to the primer, we are now using here the RNA. The DNA polymerase I would remove the RNA and transform and attach DNA polymers, and it will now fill the gaps. While in the case of the DNA ligase, it is now responsible for the connection of the complementary strand that is simply created. They will now join the ends of DNA segments and they are responsible for DNA repair. Remember, as I mentioned earlier, there is the term known as the Okazaki Fragments. This is actually a fragment of the lagging strand, and you can observe here on the part of the DNA ligase, if you are following my cursor, that there is a presence of a gap. The DNA ligase will now attach the end of this Okazaki fragment to the other end of another Okazaki fragment, so that it will now create a continuous strand. The DNA ligase will now be responsible for fixing the complementary strands that were created, specifically on the part of the lagging strand. Okay? Once again, the lagging strand is present because of the antiparallel nature of the DNA replication. But do not worry, I am going to incorporate additional video discussions when it comes to this. I am just scratching somewhat the surface, but the idea regarding this particular matter on how it is being done, I'm going to provide additional video presentations regarding this matter. In the case of the formation of the new complementary strand, remember that it is now the presence of the following. It will start with the presence of the primase, which would also be known as the initiation. Then, when it comes to the action of the primase, it will now attach here the RNA primers. Then, the presence of DNA polymerase III, it will now synthesize DNA. Then, DNA polymerase I will remove the RNA primer and create now the DNA counterpart of these primers. And lastly, the DNA ligase will now fix. Okay? DNA ligase will now join the ends of the DNA segments, specifically the Okazaki fragments. Okay, so those are actually the different proteins and enzymes that are responsible for DNA replication. And all of this are actually present on the known as the zone of the replication fork or the zone of replication. In the case of the replication fork, it is now actually the time in which all of those proteins and enzymes, it is now attached on a specific part of the DNA. Okay, these different types of proteins will now be responsible for the creation of a new complementary strand. Okay, and usually the replication will actually happen during the cell cycle, during the early parts of the cell cycle, since we need the presence of identical DNA when it comes to the presence of the daughter cells that would be responsible now for the growth of every single one of us. This is actually a complex that we can actually observe during DNA replication. You can observe here that the lagging strand will have a template that is running from 5' to 3' direction. The creation of the complementary strand will start on the 5' end and end on the 3' end. Okay, so basically if we're going to observe here, okay, follow the cursor once again. Our template, it is running from 5' to 3' direction. Then, the primer would be presented on the 3' end of the template. Since it is now the 3' end, its anti-parallel side would be running from the 3' from the left to the right to 5'. Since the primer would now be present in this particular side, if we are going to flip this from the template to the complementary strand, it will now be running from a 5' to 3' direction. Okay, that is the reason why it is now considered as the leading strand. While in the case of the lagging strand, it is now having a template of 3' to 5' direction. Since the 5' is the end, and if we're going to attach the primer here, it would now be running from 3' to 5' direction. Since it will now start on a 3' direction, the DNA should actually move a little bit forward so that the RNA primer could be attached on the 5' end. Then, the synthesis when it comes to the DNA polymerase would now be moving on a separate direction. Okay? It is now moving on another direction, not following the direction of the replication fork. If, okay? Please take note of this. If the movement of the DNA polymerase tree is not following the direction of the replication fork, it is now creating the lagging strand. Okay? If it is in the opposite direction of the replication fork, it is creating the lagging strand. But if the DNA polymerase tree is following the direction of the replication fork, it is now creating the leading strand. Okay, so please take note that it is not always perfect when it comes to the concept of DNA replication. In some cases, the DNA would be exposed to different types of external agents known as mutagens, and these agents can actually alter and create mutations when it comes to that certain type of DNA strand. And those different types of mutations could lead to different types of sickness and also could damage the DNA, the genetic makeup, and even the different proteins that would simply be created.